Hello, hello, everybody. Hello. Hello to you all. Welcome to the stream, everybody. Your boys got COVID. Let's go. Hello, John. Hello, Kevin. Hello, icosahedron. Hello, Tanith. Or, I'm sorry, however you pronounce your name. Uh, Hi, Resta. Hi, Resta. Welcome, welcome. Future collab partner Resta and Clockwork. Hello, hello. You have all come to gather around me, who has finally gotten a case of COVID. And as you can tell by my voice, it's not a tremendously serious case of COVID. I can still talk. But boy, as the night drags longer, my hubris is coming to get me. So last night I came down with the worst headache I've had in a very, very long time. And I thought that was just because I'm a headache getter. As a kid, I used to get migraines. I grew out of that. But, you know, I experience headaches every once in a while. And last night I had one of the worst headaches I've ever had and went to bed at 9 and thought nothing of it. Today I woke up with a sore throat and no other symptoms. And then Toxo prompted me to take a COVID test, and I was like, nah, it can't be. I mean, I have no fever, I have no other symptoms, just a sore throat. But lo and behold, Toxo is always right. I do, in fact, have the vid. Welcome, Natalie. Welcome. Welcome, welcome. Today, what we are doing is... So, whenever I have a condition that, say, impairs the mind, what I like to do is immediately try to test how much of my mental capacity is still intact. So as an example, at my friend's bachelor party, I wanted to challenge myself to, for example, play ping pong while drunk. So I'm very good at ping pong, and the rule at the bachelor party was for every match that I won, that I would take another drink. The issue with that is that I'm not saying that I have great motor coordination while I'm drunk. What I am saying is that I retain a lot of fine ping pong skills when I'm drunk. I ended up never losing, and the night ended very badly. That's been kind of a recurring theme my entire life. I've always been someone who, if I've had some alcohol, one of my favorite things to do is to go out and like do math problems or something, just to see what percent of the noggin is still working. And right now, while I woke up mostly asymptomatic, I am feeling quite fatigued. So it's going to be fun to try to disambiguate some of these COVID papers. I'm a little... There we go. Let's get in that corner. Okay, so what are we doing today? I've picked out a couple of open access COVID papers that we can go through together, and I will actually perform a live reading of the paper that the chat had picked. So we're going to settle on this one. I haven't read any of these ahead of time, so we're actually going to be reading this together live for the first time. And so this will demonstrate to you how I will handle reading a paper that I know nothing about. So fun fact, I actually don't know a lot about COVID, despite being a biologist. In fact, during that entire period of my life, I had purposely avoided learning anything about COVID. At first, it was because, well, the first batch of research that comes out during the pandemic is not going to be reliable and reproducible anyway. But then I just continued to ignore the science because I didn't want to think about COVID. Wasn't good for the old mental, you know? Okay, so today we're going to go through this together, and feel free to ask any questions as we go through this together, but the draw of this is, let's see how well my brain is functioning right now. Okay, so let's start with this paper. Human coronavirus NL63-NSP1 induces degradation of RNA polymerase II to inhibit host protein synthesis. So this is from the Department of Microbiology and Immunology at the University of Rochester Medical Center in the good old US of A. Very cool. Oh, that one Kyrgyzstan video. So I actually stopped watching Kyrgyzstan entirely when I started my channel because it's kind of petty and weird, but since we do operate in a similar space, I tried not to watch them, weirdly enough, just because I'm like, I don't want like to have a good idea and then learn that Kyrgyzstan already did it. It's kind of petty, but it's true. Are you doing this now because if you interpret the paper incorrectly, you can blame the COVID? I'm not saying that that's not true. Clockwork says same. Yeah, I don't watch other, like I stopped watching SciShow and John Green, and which, you know, is content that I used to really love, but now that we operate in the same space, I'm like, I don't want my ideas to have been second, so I can claim ignorance and say that, well, I never watched their video. So if there's any similarity, that's just coincidence. Okay, so let's get into this abstract. Coronavirus, COV, non-structural protein 1, NSP1. So the thing about biology papers is we're going to be seeing a lot of acronyms, and so, yeah. I had a whole thing about tuberculosis, and Kyrgyzstan completely crushed it. I mean, yes. Which is why part of my urgency in getting the Prion video out is Kyrgyzstan hasn't made a Prion video yet, and I wanted to be the first. Which is petty, but again, I wanted to get there before people started to accuse me of copying Kyrgyzstan. Consider a pathogenic factor due to the ability to inhibit host antiviral responses by inducing general shutoff of host protein synthesis. Okay, so strong first sentence. So we have a particular method by which the COVID virus can stop us from making proteins, and it's through this protein NSP1. This is expressed by alpha and beta COVIDs. I have no idea what alpha and beta COVIDs are, but sure. But its functions and strategies to induce host shutoff are not fully elucidated. Great, so that's the knowledge gap that we're trying to fill here. We compare the NSP1s from two beta COVIDs, SARS-CoV, so this is SARS and then SARS-CoV-2, so coronavirus, COVID, what we're familiar with from the last pandemic, and two alpha COVIDs, NL63 and 229E, never heard of them, and found that NL63, non-structural protein, has the strongest shutoff activity. Interesting. I wonder what relevance that has later. Unlike SARS-CoV, non-structural proteins which bind to 40S ribosomes and block translation of cellular mRNA, NL63-NSP1 did not inhibit translation of mRNA transfected into cells. Instead, NL63-NSP1 located to the nucleus and specifically inhibited transcription of genes under an RNA polymerase promoter. Okay, so this is already quite interesting. All of these viruses have the same sort of family, and yet they evolved the non-structural protein to do slightly different things. So in the end, they both stop protein from being made. What proteins are going to be made, I don't really know. Maybe just a general inhibition of host protein synthesis might prevent us from making immune molecules, which would be quite useful for the virus. But actually, even within the same family of COVIDs, coronaviruses, it appears that some are doing it by blocking the ribosome, which is the RNA protein complex that helps you make protein, and then others are inhibiting the process by which we turn DNA into mRNA, which, of course, I've talked about the central dogma over and over again, you need to eventually do to make proteins. So that's kind of interesting. Hair Day, what makes a protein non-structural? I have no idea. It's a very good question. My hypothesis, being not a COVID expert, is that there are structural proteins that make up the capsid of the protein of the virus, so things that are needed to package the virus, and maybe this is some other protein that the host is forced to make, is my guess. Good question, though. Further analysis revealed that NL63-NSP1 induces degradation of the largest subunit of RNA polymerase, RPB1. That's also quite interesting. So it's not just blocking, but it's actually sending that part of the protein factory to be destroyed. This degradation was detected regardless of the phosphorylation state of RBP1. Not really sure why that's relevant for now. I'm pretty sure we're going to learn. And was blocked by the proteasome inhibitor MG132. So the proteasome is a part of the cell that's like this garbage disposal for proteins. Okay, so we're getting hints as to how this is happening. Did I have caffeine before this? No, I would like to sleep tonight, and I also am very sensitive to caffeine. I'm just trying to get through the abstract because the paper is very long. By the way, we might not finish this paper just because if I start feeling really bad from the COVID, I might just call it a night. But I am trying to keep pace with getting into this paper. We also found that RPB1 was ubiquinated in NL63 infected cells, an inhibition of ubiquination by a ubiquitin activating enzyme inhibitor TAC243 prevented degradation of RBP1 in virus infected cells. Okay. Each time I read an acronym, I forget what it's supposed to mean. That is the life of every biologist. Eventually, you just stop caring and then you remember I can always relook it up and control F it later. I don't know how people did this before control F in computers, but yeah, a lot of acronyms. I'll try to summarize as I go along. So basically, we found, or sorry, not we, but the scientists found that this subunit of the ribosome, the protein factory, was tagged with a tag called ubiquitin. Ubiquitin is a tag that gets put onto proteins that are destined for destruction. So it actually helps proteins that are meant to be chewed up find their way to the proteasome, which is the garbage disposal of the cell. And so they know this because when they use a ubiquitin activating enzyme inhibitor, TAC243, it stopped the degradation of that ribosome. So these COVID viruses actually will destroy the ribosome by sending the ribosome to the shadow realm, or in this case, the proteasome. These data reveal an unrecognized strategy of host shutoff by human alpha COVID NL63. So this does not appear to be the COVID that we care about. Interesting. So this is like a COVID, the COVID that we are colloquially familiar with, one of their cousins, it looks like, targeting host transcription by inducing RBP1 degradation to prevent host protein expression. So I've been misspeaking for a bit. RBP1 is not the ribosome, but is the RNA polymerase. Sorry. Already I'm feeling the effects of the COVID brain fog. I'm not going to say I have brain fog, but I am a little fatigued. Okay, so it prevents the host from making the mRNA to make a protein by sending the RNA polymerase to the shadow realm. Okay, we got that. Our study indicates that viruses within the same family can use completely distinct mechanisms to regulate host antiviral responses. Hey, that's what I thought was interesting when I read the first sentence, and one of the first sentences in the abstract is that even COVID cousins have very, very, very different strategies to screw with host cells. So that's very neat. I'm not going to read the author's summary because we already read the abstract. That just helps us get right into the story. So the story of this is we're going to learn a little more about how this particular relative of the COVID virus that we're familiar with stops us from making protein by stopping us from making mRNA. Okay. I pray that someone tags me with the ubiquitin. I don't think it's going to be a very pleasant process for you. If you look up pictures of the proteasome, it's like a sarlacc pit. It's gnarly. You get chewed up in there. It's also very cool. Okay, introduction. Virus infection triggers a wide range of host defense machinery, such as innate immune responses and inflammation. It's your boy. Innate immune responses, that's me. Many viruses express proteins to counteract host antiviral activity and immune responses. True. One of the major viral strategies to regulate host responses is to induce general shutoff of host protein synthesis. Okay, so we stop making protein, we can no longer fight the virus. That makes sense to me. This is observed in many viruses, including respiratory RNA viruses such as influenza A virus and coronaviruses, COVIDs. COVIDs represent a large family of RNA viruses that infect various animals and humans. They occasionally cross species barriers to cause epidemics or pandemics. So far, 7 COVIDs are known to infect humans, 2 alpha COVIDs, and then these 2, and 2 beta COVIDs, these 2, cause mild respiratory infection and circulate continuously in human populations. Severe acute respiratory syndrome COVID, SARS-CoV-1 and 2, and Middle East respiratory syndrome COVID, which belongs to the beta COVID genus, cause more serious respiratory diseases. Okay, that's good and fair. So the one that we're interested in is one of these mild ones. Both alpha and beta COVIDs express non-structural protein, NSP1, which is known to control antiviral responses by globally reducing host protein expression. Again, so it's rehashing what we've already talked about. These COVID viruses stop us from making protein. It is considered to be a major virulence factor since NSP1 deletions of SARS-CoV-1 are highly attenuated in mice. So if you remove the COVID's ability to make this non-structural protein, you don't get as severe of an infection in mice. Okay, so that makes sense as well. Interestingly, this non-structural protein is one of the least conserved non-structural proteins in COVIDs. Okay, so that's interesting too. So, I'm going to poll chat so that we can keep this somewhat interactive. Do you know what conserved means? Yes, no. I just kind of want to see where our general knowledge is at, to see how deeply I should talk about the idea of conservation in a molecular biology context. Yeah. So I want to hang on that because that to me is pretty interesting. This idea of this particular protein not expressing not having is the least one of the least conserved proteins. I find that to be curious. Okay, so it looks like it's about half-half. So I'll go into it just a little bit. So when a molecular biologist says conservation, it means something very different than how an ecologist would see the same thing, right? So when I say, or when the authors say that it is the least conserved non-structural proteins, it means that these COVID viruses all have some relationship to one another, right? They're cousins, or in some cases, direct siblings. They are all related to each other and are all quite similar in many aspects. What this means is that the non-structural protein, the protein that we care about today, is not the same, not exactly the same across the different family members. So it's sort of unique to each strain of these coronaviruses. So this might indicate that it can do various different machinery things, depending on which virus makes this non-structural protein. And I find that to be kind of interesting. Because I think in the abstract, and I might be wrong, but I think in the abstract, different SARS-CoVs, they're non-structural proteins. Some of the family members have that inhibit the RNA polymerase, and some have them inhibit the ribosome, which are two very, 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 very different systems or structures. And so it's interesting that the same protein across different family members have evolved to do slightly different things, which is kind of neat. Okay. Although the sequence is not highly conserved, the N-terminal globular core of the COVID non-structural protein show structural similarities. Okay, so the sequence is referring to the sequence of amino acids, and if you've seen my protein stream done with clockwork, you would know what that means. Proteins are made of strings of amino acids, and so that string, that order, is what is called the sequence. And so even though the exact amino acid oh gosh the fatigue is setting in. The exact amino acid recipes for each protein, each of these non-structural proteins, are slightly different, or in this case, quite different across the different COVIDs, but their shape ends up being about the same, or at least this part of the protein, the N-terminal globular core. They all contain 6 antiparallel beta strands and 2 alpha helices. Okay, so that's just giving us some structural information. Staying up till 2 a.m. just so I can fall asleep to fire reading a document. Well, hopefully this won't plague your dreams thinking about COVID. Fun fact, I used to make fire read me bland Wikipedia articles to fall asleep to. Yeah, we used to do that quite a lot, didn't we? Caught COVID bad, March 10th, 2020. IDK what sucked more. On one hand, 5 days of 103 fever, geez. On the other, I was immune before the lockdowns even started, but had my life shut down anyway. Yeah, it was a bad time that I would like to collectively forget. So they all contain 6 antiparallel beta strands and 2 alpha helices. Okay, so structural information, the 6 beta strands that form a beta barrel fold are considered as a common fold in alpha and beta COVID non-structural proteins. So, okay, so this is just stuff telling us about the actual structure. If we don't have a picture, we don't really have to read through all of that. Okay, but this particular strain of COVID virus, this NL63, that's the stuff that we care about. So while beta COVIDs have an additional C-terminal domain, okay, so it's got an extra piece compared to its family members. And this extra piece varies in size between the 4 lineages. Okay, interestingly, studies of this particular protein from the COVIDs indicate that these non-structural proteins use their extra cytoplasmic C-terminal domain to stop translation by blocking the mRNA entry site. Okay, so this is talking about the other strains that block ribosome stuff. Although blocking translation is its primary approach to induce host shutoff, SARS-CoV non-structural protein has also been shown to induce mRNA degradation, disrupt mRNA export machinery. Yeah, okay. So main strategy, stop us from making protein by blocking the ribosome, but also it can screw with our mRNA, which is pretty interesting. Interesting. If there are conserved structural elements of non-structural protein, it sounds like the amino acid variation might be the result of synonymous mutations. That is a possibility, Resta. I think that can be true. However, so when you say synonymous mutation, what I read that as is what is it called? Not missense or nonsense, but the third kind of mutation, where you can mutate a genetic sequence but end up with the same amino acid. This is actually a great little segue, not segue, but a little pivot, Resta, that you've allowed me to talk about. So we have four nucleotides A, G, C, and T, and that codes for 20 amino acids. Each amino acid is coded by 3 of these nucleotides, so each codon is comprised of a group of 3 nucleotides. But there's degeneracy in the amino acid code, not that kind of degeneracy, but degeneracy in the way that you can have redundancy in the table that converts DNA into protein. And so sometimes you can get a mutation in the gene that doesn't affect the protein at all, which can definitely be true in this case. However, silent mutation. Thank you so much. Yes, silent mutation. Thank you. It's also possible that the amino acid sequence is very different. Sorry, Resta. I think I misread your sentence. It sounds like amino acid variation. Yeah. So amino acid variation. So it already allows you've already stated that the amino acid changes are in the protein. So even though the composition of the proteins are different, you could still end up with similar electrical, spatial information that allows the protein to fold in similar enough ways, which is pretty interesting. Okay. In contrast to the study on beta COVID, non-structural proteins, little is known about the mechanism of shut-off introduced by alpha COVIDs, okay? So I got that last part wrong. Importantly, alpha COVIDs lack the C-terminal domain. Okay, so that's actually where we care about, the ones that don't have the C-terminal domain. Here we report that the non-structural protein, by the way, I hope you get an appreciation for I do a lot of reading for these Find the Neutrophil videos, right? And I do a lot of paper reading because it's not that I don't trust science journalism, right? Obviously there are a lot of great science journalists who do great work translating groundbreaking research for non-expert audiences. But if you want the real stuff, if you want everything, then you have to read these. And they're quite difficult. I'm probably going to make a lot of mistakes interpreting a lot of stuff. You can actually see the work that goes into understanding one of these things. Okay, so the non-structural proteins that we care about don't have the C-terminal domain. So what's interesting here is that it's going to act in a different way than stopping the ribosome. Here we report that the non-structural protein from this particular COVID virus shows the strongest shut-off activity as determined by a reporter gene assay. Okay, further analysis revealed that this particular non-structural protein takes a completely different approach than the other relatives to induced host shut-off, again, rehashing what we've seen in the abstract. NL63 is translocated to the nucleus and induces ubiquination and degradation of the host RNA polymerase to reduce host transcription. This is the first report that shows that a human COVID targets RNA polymerase to induce host shutoff and block host antiviral responses. Okay, so I'm going to take a break here, and I'm going to ask if that makes sense. If the idea of the paper makes sense, because I kind of fumbled through, because again, it's my first reading. But I want to, before we get into the results, kind of catch up with you all to just kind of see where we're at in terms of, do we get the gist of this? The gist of this paper being, there's this particular COVID virus, and what it does is that it stops protein from being made, but it does so in a different way than its relatives, despite using the same protein, just a slightly different version. Okay, it looks like we got the go-ahead, basically, from chat to start diving into the results. Great. Okay. So we start with the results section, phenotypic differences between human COVID non-structural proteins. Okay. Now, if I'm being honest, chat, this is not how I would read the paper. When I do a reading for Find the Neutrophil videos, I will actually go straight to the data. I don't read any of this. I just skip right here and start reading the data. But because I am doing this for an audience, and because it would be incomprehensible if I jumped straight to the data, I will go through the author's words. And so why might that be one of the ways that you could read a paper if you have a little bit of experience? Well, the thing is my advisor, my thesis advisor, when I was in graduate school, had always advised me to never drink the author's Kool-Aid. It's basically her way of saying that the authors want you to believe and agree with them. And so by reading their words, you can introduce a level of bias. Whereas if you jump straight to reading the data, then you don't really have to be worried about getting a source of bias from the authors. Now, I will eventually read what the authors have to say when I have difficulty interpreting their data, so it's not like I just ignore it, but I do this in a slightly different order. Looking at the data with my own eyes without author interpretation is the cleanest way I can critically evaluate whether or not I agree with their interpretation. Okay, but since this is for an audience, let's read the paper in order. All human COVIDs express this non-structural protein, which was considered to play a significant role in regulating host immune responses. Okay, we know that. However, differences in the non-structural protein activity and mechanism of action of various COVIDs have not been fully elucidated. Okay, so mysterious protein doing different things in different COVIDs. Because alpha COVID lacks the domain known to be required for host shutoff by SARS-CoV and SP1s, we first compared the shutoff activity of two beta COVIDs, so SARS-CoV-1 and 2. So these are the scary ones that we are most familiar with, and these two less scary ones. The amino acid sequences of the non-structural proteins are provided in supplemental figure, which I won't go to. Maybe that'll be fun. So we can actually see. Give me a second. This paper's so long. This paper's so long. Oh, right, S1. Which supplemental figure? I lost it. Maybe we'll come back to it eventually. I wanted to look at the differences in amino acid sequence. Look at all these fun, exciting data figures that we have to get to after we read through everything, of course. Abstracted figures to download supplementary figures, PDB data if applicable, results, discussion, everything else. Yeah, that's true. My professor taught us to read the intro, then methods to decide the legitimacy. Yeah, that's fair. I tend to not read the methods until I need to, which is maybe because you will hear how they did things, and I only read the methods when I activate critical thinking. First, I'm just understanding. I'm not like in critical thinking mode yet. I'm just trying to understand. But when I'm in critical thinking mode, then I'll go into the methods. Okay. So scary COVIDs, SARS and the COVID that we know and love, and the two weaker COVIDs, we're comparing them, this particular protein. The amino acid sequences are provided. We can't get that supplementary figure, so whatever. To elucidate which stage of host protein expression this protein disrupts, we transfected 293 T cells. I forget what these are. If someone in chat wants to look these up for me, I've used them before. There's some mammalian something or another, but I don't actually remember what these cells are, which is embarrassing because they're a very common cell type. With in vitro transcribed capped and polyadenylated mRNA coding HA tagged. Whoa, that's human embryonic kidney. Thank you so much. Thank you so much. Yeah, so human embryonic kidney cells. So these are not in mice. These are not in a non-relevant population. This is a line of human cells. So that's good to know that it has some relevancy. But right now, learning that this is in a kidney cell, I am struck by whether or not this will have validity down the line, right? Because if we're talking about the relevant cell populations that a respiratory virus infects, wouldn't we want to do something like, something closer to a lung? I don't know. I'm not an expert, but that is sort of like, okay. Kidney cell for a respiratory virus. Okay. So in vitro transcribed capped and polyadenylated mRNA encoding HA tagged NSP. So just to translate that into English, so they're forcing these kidney cells to make this tagged structural protein from the COVID. All right, so this is not infecting human cells. This is forcing human cells to make a tagged version of this non-structural protein, along with two types of luciferase. So luciferase is an enzyme that cleaves a substrate and produces light. So this is how we are going to measure something. So plasmid DNA encoding firefly luciferase. Okay. So this is just the genes that you need to make the luciferase, and mRNA encoding vanilla luciferase RenLuc. Okay. So this is how they are going to be measuring. They're going to be measuring light coming off of these chemicals. If that doesn't make sense to you, that's fine. We'll get into the data later. I don't fully understand it myself, but we'll see. We then measured the luciferase activities. NSP from the scary COVIDs reduced the expression of both of the luciferases, as is expected for a host shut-off protein that inhibits translation. Oh, okay, I see. So here's their system. I get it now. We want to know whether or not the host can make protein, right? And so we could just pick a protein that the cell would normally make, right? But in this system, we are forcing the cell to make this viral protein. We're also forcing the cell to make this artificial protein that it normally doesn't make. So I actually kind of like this, because you never really know if a change in protein producing machinery is going to be because you've forced it to make something, and then inherent genetic circuits are downregulating everything else. But in this completely artificial system, I actually like the usage of an artificial way to measure protein production. So imagine this. You compare the cell that is making the COVID viral protein, and then ask it to make this luciferase enzyme. And then you compare that with a cell that was not asked to make the COVID protein. And then you can compare to see who makes more luciferase enzyme. I do like that. I think that's neat. 293T are generally just used as an expression platform because they're engineered to have really good transfection efficiency. That's fair. Yeah, I mean, that's why they've also have a place in my lab, although I haven't used them in a long time. But in terms of whether or not this happens in a relevant cell population, that's kind of where I'm like, I hope they move into something more relevant for real life later in the paper. If not, I'm not going to be too mad. It's still interesting, but it is a little like, kind of moment. Okay. This is kind of interesting because it sounds like there are some biology professionals, or at least very advanced biology students in chat, and you're going to come up to see me struggle with some of this, probably. Okay. So where were we? So the NSPs from the scary COVIDs reduced the expression of both of these luciferases. I'm not entirely sure why they used two different luciferases. Maybe we'll find out later. And then the alpha COVID 229E did not show shutoff activity. So alpha COVID 229E doesn't work this way. While NL63 demonstrated the strongest inhibition of this particular luciferase expression from cDNA, even with the less NSP1 expression compared to SARS-CoV. Okay, interesting. So it looks like their non-structural proteins might be stronger than the other scarier COVIDs, which is neat, I think. And it also looks like we're talking about two different experiments. One where you ask the cell to make the protein, and then another where you transfect it, where you infect the cells with the relevant viruses. This might make more sense when we look at the data. I'm just trying to interpret what it is we're about to see. Interestingly, NL63, blah, blah, blah. Okay, this could be due to transfected mRNA having better access to ribosomes if cellular mRNAs are shut down prior to reaching the ribosome. Don't understand that right now. Maybe it'll come to me later. These results indicate variation in NSP1 shut-off activities amongst the COVIDs, and that NL63 NSP does not block translation of captain polyadenylated mRNA transduction to cells. Okay. Okay, so that was just figures 1b and 1a. So let's look at that first before we move into 1c and 1d. Okay, so we have some figures, phenotypic differences between COVID and NSP1. So here we have a comparison between the various viruses that we're using here. And this looks to be the sequence of the non-structural proteins across the different COVIDs. So SARS-CoV-2, hey, that's the one that's in me right now. And we're comparing that to this weaker COVID, NL63, and this seemingly even weaker COVID, 229E. Ooh, so the two weaker COVIDs, despite being a part of the same family, only have 61% sequence identity. That's not a lot of sequence identity. So they only share 61% of the sequence of this protein in common, despite being close family members. That's kind of nuts. Here's the scary COVID with the extra C-terminal domain. So that's as we expected from the intro. Okay, and then B, this is what we are interested in. So let's zoom in here. See if we can't zoom in just for this figure. Get a better look-see. Before we do, though, let's read the caption. Caption is also very important to figure out what it is we're looking at. Okay, B. 293 cells were transfected with the luciferase constructs in vitro synthesized. Okay, so these two. So a plasmid. I see. So this one is forcing the cell to make this luciferase from a plasmid. And then this one is forcing an in vitro synthesized mRNA for the GFP or indicated SP for 16 hours. Here's where I'm going to struggle. Where my brain is starting to fry. And so here's where chat might be useful if there are biologists in chat that want to help. Or you can watch me struggle. That's content, baby. That's content, baby. Okay. So if we look at the data, we have two different luciferases. So some of these got a cDNA, and some of these got an mRNA. Okay, so this is relevant because we're trying to see at what stage we are preventing protein from being made. So if you get the DNA, great, okay, didn't have to struggle for too long. Okay. Here's why we do the luciferase thing twice, and this is something that I just missed, right? You see up here, and now we can actually zoom in. See up here. There you go. We do the luciferase thing twice. And so if you don't understand what this is, we are forcing the cell to make the enzyme luciferase. This enzyme is going to cleave a molecule that produces light, which is why the y-axis here is luminescence, log 10. So this is a logarithmic scale, so the difference between 3 and 4 is tenfold. So while this may look linear, there is actually quite a big difference when you see a difference, at least the light level, right? And so we do this twice because this is giving the cell DNA, and this over here is giving the cell RNA. So this is useful because the DNA has to be turned into RNA and then turned into protein, whereas here you skip that first step. So when we're trying to understand how these particular COVID viruses stop protein production, we want to know at what step that actually happens. So now, assuming that I've gotten that right, we can interpret the data. Great. Okay, so GFP. This is just our control. So this is not asking the cell to make a COVID virus, but just make a protein, and GFP is a great protein to just have a cell make. And so you get some fluorescence here. But once you make the cell make SARS, we'll just call this one SARS because that's what this one is, the scary SARS that predated COVID-2. And you see that quenches, it quenches the light, so it prevents this luciferase enzyme from being made. And the difference here looks like to be about tenfold. So there's tenfold less signal when you make the cell make SARS protein or infect with SARS. The COVID that I am very familiar inside and out with also does the same, prevents that protein from being made. And then 229E, as we saw in the body of the work, doesn't seem to do that at all. And then NL63, which seems to be the star of the show, that is way lower here. Okay, so that's interesting. And then we're going to look at the mRNA, and then what we're going to do is I'm going to ask chat to tell me why this is interesting. And I think this is a fun interactive way to walk through this kind of paper. Luciferase feels like a name that's got to have a dumb biologist reason. Oh no, luciferase I think has a great reason. So so luciferase, the root, the prefix of that comes from light, and I think Lucifer is canonically known as the light bringer, right? Correct me if I'm wrong. And so because this enzyme has the ability to produce light, it's called luciferase. I don't know if it's actually after just the LUC part or Lucifer, but it's cool. I think that's a very cool. It's better named than immune proteins, at least, with all the nonsense letters and numbers. Okay, so we're looking at the other panel with the mRNA now. So instead of feeding the mammalian cells the DNA, we're feeding them RNA. And so we see that the COVID viruses, the scary ones, they reduce the production of luciferase, but then the non-scary ones don't. So what does this mean? At what step does the non-scary NL63 virus work at? So I'll put up a poll just to engage chat for a little bit. What stage of protein production does NL63 work at? And then the answers will be DNA to mRNA, and then the other is mRNA to protein. Put that up as a poll. I'm curious as to what happened to 229E, even though he isn't the star of the show. Yeah, it doesn't look like 229E stops us from making protein, at least not measurable in this particular assay. Again, that's hedging. Scientists love hedging. We love to hedge. But it could be why 229E isn't as scary as the scarier COVIDs, right? It's just not very good at stopping us from making proteins. So maybe we're better able to make proteins that fight it, and so we can fight it. Whereas NL63, even though this is supposedly one of the less scary COVIDs, seems to be better at stopping us from making protein at this stage. So we're a little split in the chat in terms of what stage NL63 works at. And so I'll walk through the data again. So when we give it DNA, NL63 stops the production of protein. And when we give it mRNA, it doesn't. And so because NL63 has a lower bar here than here, we can conclude that it is more likely to be needed to go from DNA to mRNA, which makes sense given the introduction where we talked about RNA polymerase. Excuse me, you're a neutrophil ragging on immune proteins. Hey, I call it like I sees it. No bias here, scientific or otherwise. Okay, so that's pretty cool. I like that. The star of the show here today for this paper is this protein that's... Ooh, you know, chat. There's something interesting that I noted in this figure, specifically in this panel. In the ranitidine-luciferase mRNA panel. Does anybody want to take a crack at what I find interesting here? There's something a little unexpected about that data, and I don't really know if the authors address it. Can anybody tell? Your mouse doesn't show up on screen. Oh no, that sucks. I thought I had it set to show on screen. That kind of sucks. Let me see if I can change that. But the panel on the right where it says Renilla luciferase mRNAs. Dang, yeah. Take a second. Well, yeah, it says capture cursor in my OBS. So that's a bummer. Yeah, so what can I do? Maybe I could highlight on the thing. Can I draw? Yeah, I can draw. Yeah, I can draw. Nice. Okay. We've done it. We've done it. Icosahedron. It produces more than the control. The bars don't change by the same ratio or amount. That I'm less surprised by. Sometimes you just accept it for what it is, but icosahedron, you've got it. It produces more than the control. See, this bar up here with 4 asterisks indicates that there is a very, I'm going to get yelled at for this, but colloquially, the p-value between the p-value for the difference between LL63 and this control is less than 0.000 something or another. So the difference here seems to be quite real, assuming we trust the way that they did their experiment and that we trust their statistics. Again, I haven't read the methods because I'm not in critical brain mode, I'm just understanding mode. Well, that's weird. So you actually make, it looks like maybe about, I don't know, really like 3-fold more luciferase signal. I'm not going to say luciferase protein because it's not necessarily 1-to-1, but you get maybe a 3-fold increase in luciferase signal when you are infected with this alpha coronavirus. And that is weird. That's weird. That's just weird. Good job, icosahedron, for pointing out why that's weird. Maybe they'll get to it later. Okay, so that's cool. I like that. That's a easy-ish bar graph to interpret. So we can go on. Oh, omega big text. We can go on to talking about the rest of the figure. Because NL63 NSP did not inhibit translation, so translation is the process to go from mRNA to protein of this luciferase mRNA. So this confirms that we read their data correctly. Unlike SARS-CoV-1 or 2, we used a polysome fractionation assay to determine whether NL63 binds to ribosomes. Okay, so I believe a polysome fractionation assay is taking the cells and spinning the absolute crap out of them, spinning them in the centrifuge at very, very high rotations per minute to try to sediment out the ribosome, as has been reported to SARS-CoV-1. And I assume they're going to detect that protein somehow, which we'll see in the data. Consistent with other reports, SARS-CoV-2 NSP was recovered from fractions containing the ADS ribosome and ribosomal subunits. So I'll get into what that means in a little bit. In contrast, this NSP was not detected in ribosome-containing fractions, similar to SARS-CoV. Okay. We next analyzed the localization of this non-structural protein in these cells by immunofluorescence. Ooh, we're going to see some fluorescence imaging, which is very exciting. Consistent with previous reports, this protein was predominantly localized in the cytoplasm. Oh, I'm so excited to be able to look at some fluorescent data. That's going to be cool. In contrast, this particular protein was found both in the nucleus and cytoplasm in 293. This is actually going to be very relevant for the prion phybit that I'm going to release maybe on Friday or something, suggesting a function in the nucleus to induce host shutoff. Okay, cool. Okay, so very excited. Let's go see that data. Ooh, look at the pictures. So pretty. So the ones that are going to be relevant for us, so we'll look at C first. Okay, so the thing to know about ribosomes is that they have a couple of different parts. And so when you spin down the ribosomes at this very fast pace, you can start to separate those different parts. So the smallest part is the 40S subunit, then you have the 60S, and then the ADS, and then when you have multiple, I believe polysomes refer to multiple chains of ribosomes, but I could be wrong, but they have to be bigger than the ADS. That's what this is saying. So when you do this fractionation, when you separate them into these different pieces, you can then use antibodies to try to detect the protein of interest. So it looks like in the ADS fraction, you see the most of this protein of interest in all three of the samples. So it's, is that right? I don't know if I'm reading this correctly. Okay, so we'll go to the caption. 293T were asked to make transfected with the indicated cDNA. Okay, so made the protein, treated, and lysed. CHX, I'm guessing, I don't actually know what CHX is. May or may not be relevant, we'll see. Lysate was layered over a linear sucrose gradient, so centrifuge, fractions were collected, top absorbance during fractionation, bottom proteins were precipitated from each fraction. Absorbance at 254 measured during. Okay, I think this is just the quality control thing to see where that ADS is coming out. I'm not entirely familiar with this particular method, but what I can say is looking down here. So, bottom, what is that bottom? Proteins are precipitated from each fraction and analyzed by Western blot using HA antibodies. Okay, so if we look here, it looks like the wild type, so the regular COVID protein, seems to come out a lot in all of these parts of the ribosome. And then when you remove that C-terminal domain, you don't get as much of that binding. And since the NL63 doesn't have that C-terminus to begin with, it also seems to not bind very well to the ribosome. Although it is seeming like it does bind quite heavily to 40S and 60S, and I'm not entirely sure, you know, what that means, right? Because the whole idea here is that it's not supposed to bind the ribosome, so why does it bind 40S? Oh, no, it doesn't. 40S is here. 40S is in bin number 3. Okay, so bin number 3, we draw a line down here. You see that the regular COVID virus, you do get the protein binding to the smallest part of the ribosome and the larger part of the ribosomes, which are these. But if you draw a straight line down, you don't get that binding when you remove that C-terminal domain. And since this guy doesn't have that C-terminal domain, you barely see that band at all. Okay, okay, I understand. Okay, great, cool. Okay, so the protein that we care about, which doesn't stop translation, doesn't bind the ribosome, that's great. That's a good thing to learn. It's a little redundant with what we know already, but it produces more consistent evidence. So science is all about building and building and building these pieces of proof, and so this just makes sense. It doesn't bind the ribosome. It fractionates out much earlier than the relevant ribosomal subunits, so that's great. Okay, so figure D. Oh, I want to get into that so bad. Let's read the caption first so we understand. So the cells that we care about were given the mRNA expressing the protein, so not infected with the virus, but given the mRNA needed to make the virus, and were stained with an antibody against the tag that it was made with. Cell nuclei were counterstained with DAPI. So counterstaining, it's just we're staining for the nuclei as a contrast to the protein that we care about to understand where this protein is. Images were captured using the 40X objective. The signals in total and nuclear regions were quantified by image J, and the percent of the protein in the nucleus was calculated. Okay, so let's take a look at this and see. Oh, I love data like this, Chad. I love data like this. It's so pretty. Although, I don't love how out of focus these images are. they're a little fuzzy. I think the processing on these pictures could be a tiny bit better. I first remember learning about this Friedberg unit in the context of 16S and 18S rRNA and finding it really cool how it's a unit of time influenced by the physical properties of a particle. I actually don't know too much about this Friedberg unit. It's been a while since I thought about 16S and 18S rRNA. Those are the bacteria ones, correct, Resta? Uh, I'm not really a bacteria person, as you might be able to tell. Okay, pretty pictures. Let's ignore this for now so that we just, without bias, look at these pictures. So DAPI tells us where the nucleus is, and NSP1, this is our protein, this COVID protein of interest, and so these are the different family members. So chat, without looking at this, ignore this. What do you notice about the difference between these COVIDs and these COVIDs, just in this green image right here? What do you notice is different? I'll give you a second to think about that. What do you notice in this row? 16S in prokaryotes, 18S in eukaryotes. Whoops-a-doodles. Yep, haven't thought about the ribosome in a very long time. Not in any kind of meaningful depth, at least. The thing about biology is that you need to have a very good memory, but more importantly, you need to have recognition. The left, you have dark circles. Yes, hair day. That is absolutely what I am seeing here too. So I'm not looking at this data at all. I'm just seeing with my eyes. What do I see here qualitatively? What's the difference in quality here than the quality here? And these have circles in them. Great. So let's get into what these circles might mean. So we have DAPI. This blue indicates where the nuclei are. And then when we merge, it's such a shame because these are so out of focus. It's really hard to tell. But actually, hair day, you pointing out that there are circles here is quite helpful, because what we do see is that the NL363 has a much more diffuse localization. So it seems like at least what we can say, here's what we can say. Okay, this is actually a really good point. Okay, so we'll take this just by observation without any bias. Hair day pointed out that we see circles here and we don't see circles here. Now, what this tells me, the fact that we have circles here, is that this particular protein that these scary COVIDs make does not appear in the nucleus. It is excluded from the nucleus. What can we say about these two, chat? And this is a bit of a trick question. It's a bit of a trick question. I feel dumb, I spare. This is like a college level kind of reading of a very high level paper. It's okay to feel dumb. You're not dumb, by the way. You just haven't spent years studying biology. So that's what I'm here to help with. So if you are unable to follow along, no worries. No worries. We'll take it slow, one step at a time. Icosahedron. It feels like the proteins are more spread about. Yes, they're more spread about. And I love that. I love that because if you're not a biologist, then you might have a particular bias. Or if you are a biologist, you might have a particular bias when you start thinking about proteins. And the fact that you are, the fact that some of chat are not biologists is actually really good, because you don't have the initial bias that I have. Because when I see this, I see, oh, maybe it is in the nucleus. But actually, icosahedron, your interpretation is less biased. All this green is saying is that the cell just kind of makes it everywhere. They either don't have nuclei or they don't exclude the protein in question from their nuclei. Hair day, that's actually where my brain went because of my inherent bias as a biologist and because I read what the authors were saying, right? This is why my professor told me not to read the paper, because you can buy into their bias. They want us to believe that this protein is being made in the nucleus. Well, let's see, right? Can we really say that it's being made in the nucleus? We don't actually know what the rest of the cells look like because we don't have a cell shape, right? Like, what if this is the cell shape? Or say, for example, what if this is the cell shape? If this is the cell shape, the DAPI is so diffuse, it's really hard to tell where the boundaries of the cells are. Like this image, if it were crisper, if the background were lower, I'd be more understanding. But the pictures that they took, I mean, this is so big, it's not the best image. Whereas these are a lot better, right? you can see that there's not really green around here. This COVID excludes the protein from the nucleus. Here, the staining is kind of diffuse. It's not as good. But technically, you can say that the nuclei, and I'll circle one, are co-localized, meaning overlapping, meaning that this protein is potentially in the nucleus. Now, I'll buy that because it is co-localized. I just wish that the boundaries, that the image quality was a little better. Okay, so let's go ahead and erase some of that. Okay, so yeah, upon further inspection, it does look like it is in the nucleus, as Hairday had said, which means our biases were, I believe, correct. It's just that at first it's kind of hard to figure that out because of how diffuse the staining actually is. And again, I think if they had done a little bit of a better job doing background correction, I might not have initially been like, hold on, it's being made everywhere, because they just look like cells. Which is kind of interesting. Yeah. I mean, they don't have to. Like, I'm not ragging on the authors for not including another stain for morphology, because I think that would just be extra credit. I don't think that it's really worth doing that kind of thing. I just really wish that they cleaned the picture up a little bit. That's all. Just finished a pre-on video, and as someone who hasn't taken biology since the 9th grade, I can't wait to not understand anything. Well, that's not the goal here, right? The goal is to practice the art of scientific thinking. And again, this is like college level stuff. So I love the fact that there are non-biologists in the chat willing to push themselves, because I think that biology is accessible enough that anybody, if they really tried, can do this with enough background, of course. Why take such good pictures on the left and not on the right? Speaking as a former benchtop scientist, sometimes it happens. It's kind of hard to tell. Also, is there something else? Is there something else interesting about this row, chat? This row here. Oh no, where'd my pen tool go? Here we go. This row, this dappy row. Let's compare this box to this box. Is there something interesting there besides the fuzziness? My first thought was that the protein in the first two images is more prevalent near the membrane. Yeah, we don't actually know where the membrane is here because we only have the nucleus, right? So this empty circle is where the nucleus is, according to this blue line here. So the membranes could be way far out here. It's just around the nucleus. And that makes sense because that's where the rough ER is with all the ribosomes and stuff. So that makes sense to me. But there's something interesting that I want to note between here and here. Something about the morphology of these nuclei. I'm wondering what the intensity of the color looks more zoomed in on the right. Yeah, Durano. I see some darker spots in the left one. Yeah, the darkness, that's kind of a background subtraction issue. This one maybe is just a little messier. Maybe Dappy's sticking to things Dappy shouldn't be sticking to. But these look a little bigger. Now, they didn't measure the nucleus size, but as I'm reading it and as I'm looking at the data, which is why it's important to look at data, I am thinking like, are the nuclei infected by this COVID enlarged somehow? Like, that would be kind of fun, right? Down here, the white bar indicates a scale bar. So this scale bar allows us to understand what the exact distance comparison between all the images are. So there's a huge nucleus here, enormous. And granted, this is one subsection of a lot of nuclei, but the authors chose these pictures. I wonder if they are going to make a point about these weird shaped and huge looking nuclei. Kind of cool, kind of interesting. Okay. So let's look at the data. So between the two scary COVIDs, there's no difference. The percent of the protein that ends up in the nucleus. So there's some, it looks like about 20%. But our two lovely alpha COVIDs, it looks like it's about double. Double of that staining ends up in the nucleus. So quite a bit more is in the nucleus of these less scary COVIDs. Okay, so that supports this idea that this particular COVID protein, NSP1, is doing something in the nucleus, likely preventing mRNA from being made somehow. Here's another kind of skeptic, critical thinking kind of question here. So I'm going to repeat how the authors generated this data, and I want to ask if chat can kind of predict what I might say about whether or not this is real, or my faith in this data. And again, I love plus one pathogens, open access journals. I can talk about them on stream. Love them. I'm not like, I don't want to make my career to be criticizing scientists. It's just, I want to model critical thinking. So I'm going to read how this was done, and I want chat to kind of guess which part of this I have an issue with. So these cells, these kidney cells, were transfected. So they were given this mRNA expressing the COVID protein. And then we used antibodies to image them to get these fluorescence images. These cells were counterstained with DAPI, and then the pictures were taken. Okay, so there's something I have an issue with here, and it's that we gave these cells the mRNA for this particular protein, and then that cell made that protein. And then we are seeing where that protein ends up in the cell. I want to ask chat, do you think, chat, that, or how confident are you that this is reality, that the COVID virus actually puts the protein in these locations? What is a potential issue with coming to that kind of conclusion? So I'll repeat the question one more time. We asked very politely these kidney cells to make this COVID protein, and then we asked, where did that COVID protein go? Does this reflect an actual infection by a COVID virus? Why or why not? Why or why not? Using kidney cells is weird. We did talk about that, yeah. We did talk about using kidney cells as being weird. That could definitely cause issues, but I'm willing to give grace to that particular point. Of course, it would be better if they were in lung cells, but I can accept that they use kidney cells. What's another kind of weird thing that could happen? Variables around the other proteins the virus makes. Yes, good job, icosahedron. Exactly. So this is an artificial kind of system, and that's fine, right? There's no issue with having an artificial system, but an infection brings with it a lot of other different proteins that the virus can make. And so I'm not saying that this is wrong, but I'm saying when you read this data, you have to think, okay, this is in kidney cells, and we're not using an actual virus, we're just giving them the mRNA. It's not quite real, but it doesn't mean that this data is useless. It just means that it doesn't look like a real infection. It doesn't mean it's wrong. It's just, you have to keep your skeptic hat on. Did the mRNA still include the cell sorting code for the transcribed protein? I'm not entirely sure what you mean by that, Zeal. The mRNA is capable of being made into protein, if that is the concern, but icosahedron's got it. This is not a real infection with a real virus. It's an artificial system, which again, totally fine. Just, we can't take this for absolute truth quite yet. That's one attitude that I want to model for chat. It's that when you read science journalism, or when you read about science in general, there's a lot of nuance, and journalists can kind of flatten that kind of nuance. And so that's how you get a lot of R science Redditors who think they know better than scientists criticizing studies. And I don't want you to end up that way. Right. Because there's nuance. There are flaws, but no science paper is perfect. It's about an accumulation of evidence. Okay, so let's try one more figure. Let's try one more figure, and then I'm probably going to call it a night. Oh, look at this. Oh, I want to know what this is. So let's see what that is, and then we'll call it a night. This has been fun. I'm having an easier time reading a paper live than I thought. I think I was worried that I would have to do a lot more Googling, but so far this is pretty understandable for me. How fatigued do I feel? I feel alive. It's just my throat is feeling like I've been talking for a bit, which is, just true. If I can't talk the next couple days, it's not important. We canceled all our plans anyway because I tested positive, so if I lose my voice, it's going to be okay. I already recorded my Prion 5-bit too, so don't even really need my voice for that. Okay, we push on. So this protein inhibits transcription, so the process to go from DNA to mRNA by RNA polymerase II. Okay, so we determine the effect of this protein on host RNA synthesis by a de novo RNA synthesis assay measuring 5-ethinyl-uridine incorporated into transcripts. So I believe 5-ethinyl-uridine is going to be a special version of the chat. There's the DNA bases, which are A, C, G, and T, and then there are the mRNA bases, which are A, C, G, and U. C is uridine. And so I'm guessing 5-ethinyl-uridine is a special labeled version of uridine so that we can measure RNA and not DNA. Very clever, very clever. Scientists are very clever. So we're adding this special uridine to try to track RNA synthesis. So when you see the signal from 5-ethinyl-uridine, you're tracking the production of new RNA, which is cool. 293 T cells were given this non-structural protein mRNA. 6 hours later, they were labeled for 1 hour with 5-ethinyl-uridine, and newly synthesized RNAs were detected by click chemistry. Oh, that is fun. Click chemistry. Let's see how they do click chemistry when we get to the figure. As expected, scary COVIDs did not affect host RNA synthesis. We already learned that they do it by blocking the ribosome, so that makes sense. Similarly, this non-scary NSP, which has negligible shutoff activity, did not have an effect on RNA synthesis. That also makes sense. In sharp contrast, cells expressing NL61 structural proteins strongly reduced mRNA synthesis at 6 hours post-transfection. To confirm the effect, we used qPCR, so polymerase chain reaction. I'm sure you're all familiar with that if you've taken a PCR test for your COVID tests to quantify luciferase mRNA, so our protein readout in the expressing cells. No difference was between control cells with GFP and then the scary COVIDs. Okay, so what we expect pretty much. We determined whether this inhibition is specific to RNA polymerase II. Okay, so maybe we'll just do figure 2A and 2B, and then we'll call it a night because this is getting into the weeds just a bit. Pretty pictures, let's go. Okay, ooh, magenta. Love a magenta kind of figure. Okay, so what are we looking at here? So let's ignore this because we need to know what each individual piece is. So NSP1, this is our protein of interest. It's the one that is supposedly going to be blocking the production of mRNA. EU, this is not European Union, this is 5-ethinyl-uridine. So this is a labeled version of that uridine base, which allows us to track the production of mRNA. And then we got a bunch of white arrows that will look at the caption to interpret. And then we have DAPI, which measures the nuclei. Okay, so let's go ahead and go to the caption and get to the bottom as to what these white arrows are supposed to be. 293 transfected with mRNA, yada yada yada. Fixed cells were processed for click-it reactions with elexiflor, azide, followed by IF. Okay, I'm just gonna accept that works. Cells were counterstained with DAPI arrows, and arrowheads indicate cells expressing and not expressing NSP1 respectively. Indicate cells expressing and not expressing NSP1. That's a That's a bad. Whoa, that's confusing. That's bad. That's a bad way to do this. Okay. So arrows indicate cells expressing. Okay, so this cell is making NSP1. 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 Okay. And then the arrowheads are cells that are not making NSP1. Okay. Okay, I get it. Let's get that zoom in so that you can see the figure. Okay. And we'll just compare these. First we'll do this one, I think. So NL63, right? This is the star of the show. What can you tell me the difference is, chat, between the cells that are pointed to by the arrows versus the arrowheads? So this cell is got it confused again. Oh, this is such a weird way to do it. What is this? Arrows are expressing. Okay, so this is making protein that we care about, and this one is not making the protein that we care about. So what can you tell me about the difference between this cell and this cell? Just visually, by eye. You don't have to have a degree in biology to tell me what the difference in the data look like. Arrowheads are brighter, correct. Okay, so the arrowheads are pointing to cells with more of this red signal. So the red signal comes from the incorporation of 5-ethinyluridine, which means that there is more mRNA being made. So I'll write some statements, and then chat, you can tell me the conclusion. Arrows, cells making COVID protein. Arrowheads, cells not making COVID protein. Red signal is equal to mRNA being made. Conclusion equals question mark. What do we conclude from this data in our COVID of interest? What's the conclusion here? And again, this is one of those instances where you don't have to have a degree in biology to be able to read this kind of data. Correct. Archie, I'm guessing. Archie is correct. The arrows point to low background or low signal, and then the arrowheads point to brighter. And the red signal corresponds to the amount of mRNA being made. What does that say about the cells that make COVID protein? Maybe I'll add that as a prompt. Cells that can make COVID protein make high or low mRNA. Maybe I'll do that. High versus low mRNA. Just to check that y'all are following along here. If the cells that are making the COVID protein, low mRNA, that's one of the answers. It's blocking translation. So translation is mRNA to protein. We're talking just measuring mRNA now. Low mRNA, yeah. So that's a technique that I like to do in class when students are reluctant to participate, is allow them a lower stakes version. Lower stakes being the poll. Yep, blocking transcription. Yep. Transcription is DNA to mRNA. So it looks like in our strain of interest that this particular protein blocks mRNA production. Let's look at the COVID that I have. So same deal. Arrows point to cells that are making the COVID protein. Arrowheads are pointing to the cells that are not making the COVID protein. So do we see a difference here, chat? Do we see a difference here? In the COVID that I currently have, so I should word this a different way. I'm infected with this COVID right now. Based on this picture, is my mRNA production being inhibited right now? Is my mRNA production being inhibited? It's kind of fun. Is my mRNA being inhibited? The last poll that we talked about, that was for the star of the show, alpha COVID, and now we're looking at the COVID that I have. What do we think, chat? All right, half of you have gotten an answer in. Yeah, that's correct. The answer is no. My mRNA is not being blocked here. Let's compare the two. Here it's very obvious. If you're being, if you don't get it, the arrowhead system is really confusing. I don't really like it. But this cell is making the protein. It's stopping mRNA from being made, which is why there's no red signal here. These cells are making the reversed. This cell is making the protein, so it's low mRNA signal. This cell, I can't do this, and that. Oh, I'm sorry that I have the cursor doesn't actually. The cursor doesn't appear. I keep on forgetting that. I need to draw. So I'll draw again. Okay. So this cell is deep red because the cell is not making the protein. This cell is making the protein, so the mRNA is very low. This is in the weaker COVID strain, the alpha COVID. This is the COVID that I have. So this cell is making the protein, and this cell is not making the protein. And they're about the same. The difference here is not very big. It's kind of hard to tell. You have to kind of, it's a little subtle, I should say, right? Like everything is red. If you don't know what you're looking for, then it's not entirely obvious. But these arrowheads are much brighter than these, which are just the arrows. Okay. All right. And so that's exactly what we see here, reflected in the data. And then what I want to do, before I call it a stream, I just want to double check the nuclei. So icosahedron had mentioned that the nuclei do look kind of bigger in the NL63 COVID. And I want to just kind of see if that's still the case. So I'm going to zoom in here. So this is the COVID that I have, and then this is the COVID that we're interested in. And I'm going to put a poll to the chat real quick. Which one do you think looks bigger? Top nuclei, blue, or bottom nuclei, in blue? So an earlier figure, I was like, man, those nuclei look huge. And now I want to see in their other pictures if we can still see whether or not that's true. Which look bigger? Yeah, chat's saying that the top nuclei look bigger. Yeah, it was the opposite in the other figure. So I think like in this figure, the COVID that I have, the nuclei look bigger. In a previous figure, it was the opposite way. So I think that like difference in nuclei size was just a coincidence of picking one picture to be representative. So that's a good check for just kind of like being observant, but not like getting married to your earlier observation. So I can walk back that idea, right? Like I don't, if you love your ideas too much, if you're like, oh, wouldn't it be cool if NL63 enlarged the nuclei, then you could end up being wrong a lot of the time. And it looks like I was wrong, which is fine. But again, this is how someone would critically look at the data. I think they had problems with zooming in and focusing. Yeah, like supposedly their zoom and focus should be the focus is what's the issue there. I think the zooming in, it should be the exact same zoom. But the focus they might have had issues with. So there's probably nothing freaky going on with nuclei size, probably. Okay. So my voice is getting a little hoarse. I hope you found it to be kind of interesting to at least start to walk through a scientific paper live. This is the kind of thing that my V theorem contemporary, Sidere, does on a somewhat regular basis, in terms of she'll actually go through a psychology paper live, much in the same way as I have. Although it's kind of fun because I find that my audience is very, very tuned into the actual paper. I'm able to stay very focused on the paper, and her audience just loves to derail her. It's so funny. Very worth watching if you haven't tuned into one of her streams. But thank you for keeping me company as I have suffered through a little bit of a touch of the vid. I will be taking a couple days to rest, obviously, but if I feel fine, I guess I don't know, make content or something. Definitely going to rest my voice tomorrow. But yeah, thank you all for coming out to hang out with me on this guerrilla research paper stream. I hope you have a good rest of your days. I hope you feel better soon. Thank you so much. Never had to read a paper since they just focus on cramming knowledge into our heads. Yeah, I mean, with biology, you have to know everything before you even get to this level, right? Like, I can do a good job. I can do as good of a job as I can to summarize what you need to know. But reading an actual paper, I don't know, it's kind of fun. I don't know. Before I sign off, I just kind of want to poll. Is this fun? Is this fun? Should I do more of this? Like, I mean, I guess this is selection bias, right? This is survivorship bias. Like, you've stayed to the end of the stream, so it's likely that you find it fun. I don't know. Like, it's different than a lecture because that's something that I've had time to process. And the process of troubleshooting this live, I hope it kind of humanizes science in a way, because I don't know. I was just in a meeting with a bunch of professors talking about the curriculum for our students, and I still view professors as like almost godlike. That they're so like smart. They're almost like untouchable in many ways. But at the end of the day, they're just humans too, right? They can get stuff wrong as well. I'm not a professor, but in terms of like, because professors are mysterious to me, and I'm making content in the proximity of professors, I just figured that it might be interesting for an audience to see someone work through a paper live. Yeah, I'm glad that y'all find it kind of fun. Maybe we'll pick up this paper, maybe we won't, because I think that the rest of this, because I don't want to pick this paper up later, because I'll have to re-explain everything, and then that would take like an hour. So maybe if I do one of these in the future, I'll do a paper with a main figure and then not worry about the subfigures, but like the gist of the paper, to watch someone work through a field that they're not comfortable with. All right. Yeah, I think those are interesting ideas. Yeah, brain free. Thank you so much. I will take a long rest. Bye, y'all. Whoop, wrong scene. Bye, y'all.